So if our cells have excess glucose, they can store the excess glucose as a polymer as polymers of sugar, which is called glycogen. So if there's too much sugar in a cell, too much glucose, the glucose 6-phosphate can be converted to glucose 1-phosphate. That can be polymerized into glycogen, a big polymeric sugar found in our cells that can store these glucose molecules from when they're needed in the future. And if, let's say, so after a meal is the time that you would basically, you eat a lot of carbs, then your body can store the excess glucose in glycogen. And this is why runners, the day before a marathon, they'll often have a high carb meal because they want to build their glycogen stores so when they're running the marathon the next day, that they can have enough energy for their muscles to be continuing them running this long race. So, but hey, so you're gonna, let's say, have a carb-rich meal the night before a marathon, but then you're actually doing the marathon. Well, then your glucose levels are getting depleted. So at that point, your body wants to break down the big polymers of glycogen, big glycogen polymers of sugar, and break it down. You want to so you'd break it down to glucose 1-phosphate and then convert it to glucose 6-phosphate. And then that can be used to perform glycolysis. We have one really special tissue, an organ in the body that can basically, that can help out the skeletal muscles when they deplete their glycogen stores. So, right, so the skeletal muscles are first going to use their sugars they have that are freely available. Then they're going to break down their glycogen store that they had made. But let's say they deplete that too. Well, the liver has the ability to not only store glycogen, from where we're having excess glucose, it can break it down, and then it can actually dephosphorylate glucose 6-phosphate and then secrete the glucose into the blood. Glucose can travel in the blood, get inside those skeletal muscles, and let them then perform glycolysis to continue contractions that are needed for an endurance activity. And then you can see, so most the cells in our body can basically pick up free glucose from blood. But the only organ in the body that can take glucose 6-phosphate, dephosphorylate it, and release glucose into the blood is the liver. So there's another pathway I just want to mention, which is gluconeogenesis, which is the production of glucose. So basically, this is gluconeogenesis is glycolysis running in reverse. So basically, the glycolytic intermediates can be converted in reverse reactions all the way back to glucose. I just want to mention that. So if a cell needs glucose for some kind of biosynthetic pathway, then there is a process that they can make glucose in the absence of ingesting glucose. So now I want to move on to the move on to the aerobic respiration, which is the citric acid cycle and electron transport system. And these are located in the mitochondria. And I just want to remind you that glycolysis occurs in the cytoplasm. So pyruvic acid is the end product of glycolysis. And in order to enter the citric acid cycle, there needs to be another redox reaction where NAD will become reduced, and then pyruvic acid is going to become oxidized, right? So it's going to give hydrogens to NADH plus H, and it's also going to be condensed onto this sulfur of CoA, the CoA enzyme. So what we're going to get out of this is we're going to have the acetyl-CoA being produced, and carbon dioxide will be released. And acetyl-CoA can funnel into the citric acid cycle. And this redox reaction is very important for allowing this process to continue. So again, glycolysis happens in the cytoplasm, pyruvic acid is produced, 2-per-glucose. It is then going to be transported into the mitochondria, and there we'll see the decarboxylation reaction and the redox reaction, and while NAD is going to be reduced to NADH plus H. And then we're going to have acetyl-CoA being formed, and of course CoA is going in. For that to happen, this is a 2-carbon sugar. So here's a 3-carbon molecule, right? It's not a sugar, it's a 2-carbon molecule, not a sugar. So here's a 3-carbon pyruvic acid entering the mitochondria. There's a decarboxylation reaction, so it goes carbon 3 molecule to a 2-carbon molecule, acetyl-CoA. It is going to be condensed with oxaloacetic acid, which is a 4-carbon molecule. So 4 plus 2 gives 6, yielding citric acid. There'll be a decarboxylation reaction, meaning it's going to be a 6-carbon going down to a 5-carbon molecule. There'll be another decarboxylation reaction, converting the C5 to a C4, allowing the cycle to continue. Now, in this cycle on the bottom, not shown in this slide, but will be showed in the subsequent slide, is these enzymatic reactions are going to generate 3 NADH plus H, 1 FADH2, and 1 ATP. So I already mentioned that each NADH plus H produced can go to the electron transport system and generate 2.5 ATP. And what I didn't tell you yet is each FADH2 can go to the electron transport system and generate 1.5 ATP. And then, of course, this ATP in here is being produced by substrate level phosphorylation, which is just an enzymatic reaction that's producing the ATP, whereas the conversion of NADH plus H and FADH2 into ATP, that is the process of oxidative phosphorylation. So the bulk of ATP made in our cells under aerobic conditions is produced by oxidative phosphorylation. Under fermentation conditions, right, where there's no final electron acceptor, no oxygen, then all the ATP is going to be generated by substrate level phosphorylation. And in the presence of oxygen, we get ATP from both substrate level phosphorylation, a little bit, and then a lot from the oxidative phosphorylation. So here's some more details about the Krebs cycle. And for both glycolysis and the Krebs cycle, you don't need to memorize all the enzymes and all the steps because it's just too much details for us. But what I do want to point out is that we have these redox reactions occurring. We have the NAD being reduced to NADH plus H. Again, over here, NAD being converted to NADH plus H. Here we have the FAD being reduced to FADH2. And over here, we have the NAD being reduced to NADH plus H, right? So that's where we're getting the 3 NADH plus H, the 1 FADH2, and then we're getting an ATP being generated via substrate level phosphorylation. And of course, we also have the decarboxylation reactions. We have one occurring here, another one over here. So if we look at the electron transport chain, where the NADH plus H and the FADH2 will give their electrons, we see that there's these energy level, right? So NADH plus H has greater electron energy level than FADH2. So what's going to happen is the NADH plus H will be donating its electrons to the electron transport system at a very high energy level. FADH2 will do it at a little bit lower level. And each of these is represented by a different proteins in the electron transport chain. So the NADH plus H is going to give its electrons to complex 1. FADH2 is going to give its electrons to complex 2. And the NADH plus H electrons will be going through complex 1, will be passed to complex 2, where the FADH2 electrons can come in too. Cytochromes will pass these electrons down further to other cytochromes. And eventually, the electrons will be passed to oxygen, and when the electrons / hydrogens are added to oxygen, water will be produced. So oxygen is the final electron acceptor, and that's the lowest in this relative energy level. Oxygen is going to be on the bottom of this, and it can be oxygen can be reduced to water. And what critically happens during this passing of electrons is this is where complex 1, where NADH plus H donates its electrons. Here we have the second pump where the FADH2 gives its electrons. And as the electrons are passed from complex 1, 2, and 3, what happens is that the hydrogens are going to be pumped. Basically, they're going to be pumped from the matrix of the mitochondria across the inner membrane into the space between the inner membrane and outer membrane of the mitochondria. And there's going to be a very, very high amount of protons in this intermembrane space. And this high concentration of protons in the intermembrane space has energy. It's like water on the top of a waterfall or a cliff, right? That water on top can fall down, and then when it comes down, it's going to make noise, and it can turn one of those turning devices, almost like a windmill, but a watermill. It can turn the watermill to produce energy. So this energy created by the protons being pumped across from the matrix to the inner membrane space can accumulate, and then at the end we have a ATP synthase. And this allows the protons to travel from the intermembrane space back into the mitochondrial matrix. And as this the energy of the concentration gradient is released, ATP will be generated. This is the process of oxidative phosphorylation, where the electrons are given to these electron transport system proteins. The hydrogens are going to be pumped. And then as they travel back into the matrix through the ATP synthase, ATP will be produced. Specifically, we know we get about 2.5 ATP from each NADH plus H, and we get about 1.5 ATP from each FADH2. And I am going to ask you to memorize those numbers, the 2.5 for the NADH plus H, and the 1.5 ATP for the FADH2.